students, today in this class we are going to cover the part of microbial energetics that has to deal with glycolysis, which is consumption of glucose. In the previous lecture I talked about how delta G drives any microbial reaction and decides whether a microbial redox reaction is feasible for microbe or not, whether microbe should invest energy in or not. We also talked about certain energy rich molecules within cells such as ADP, ATP, NADH, NAD+, which help catalyze these redox reactions and ensure that the energy produced from this redox reaction is essentially transferred to the cell for its essential functions such as cell replication and sustenance. In this lecture, we are going to come back to one particular important part of the previous lecture, which is that glucose is the basic substrate of most chemoorganotrophs. So, for most microbes that use organic material as their electron donor, need to break it down into glucose, which is a very simple sugar, 6-carbon sugar, and then the glucose can either be fermented or respired. Now, you must know this from your high school biology, that even in human body we have two forms of respiration. One is aerobic and one is anaerobic, which happens under extreme conditions. So, for example, a sprinter is a classical example of an athlete that uses anaerobic respiration to drive their motion. So, when the air is rich with oxygen and there is plenty of oxygen in the blood, most of our respiration and glycolysis will be governed by aerobic degradation of glucose. But when the air becomes scarce, which happens when the body is active very fast or the oxygen in the air is less, and in case of the sprinter's body is moving at a much faster rate than the oxygen in the blood can cope with, then the body of the athlete switches to anaerobic respiration and lactic acid is produced as a byproduct, which for a long time was considered to be the cause for DOMS, which is delayed onset muscle soreness. And now we know that is not the case, but we still know that after anaerobic respiration our muscles are rich in lactic acid, which give it an uneasy feeling. So today we are going to understand what this aerobic respiration and anaerobic fermentation are about, what the pathways are about. Now these pathways are very important to understand, because each of these pathways, even though in the way we will cover in this lecture is just 3 steps pathways, but they are in fact much more complicated. The web of glycolysis is so intense and so diverse also that if we were to draw this on the board behind me, it would be an impossible activity. Definitely impossible to make it legible enough for you to see. And I must mention this here, that for many decades scientists have been working very hard to uncover the glycolysis pathways and various enzymes that participate in it. So let's start today with exploring fermentation versus respiration. Now both are methods of glycolysis, which is consumption of glucose. However, fermentation happens under anaerobic condition and respiration under aerobic condition. Now in glycolysis, the cell wants to conserve energy, whether it is fermentation or respiration. So to conserve energy, it uses different mechanisms for producing ATP. In fermentation, it will do what is referred to as substrate level phosphorylation. It will directly phosphorylate the substrate, which is glucose, and thus avoid any intermediary and directly get energy from the electron donor, which in this case is glucose. In case of respiration, because the oxygen is present, this is oxidative phosphorylation. So the cell actually uses its proton motive force, and this is something we talked in one of the previous lectures, how naturally the cell membrane behaves like a battery which carries energy, which stores energy for the cell with positive charge around it and negative charge inside it. So in oxidative phosphorylation, which happens in aerobic respiration, microbes use this charge to make ATP. So when they have made ATP, the voltage around the cellular membrane reduces. So in case of glycolysis, there are usually 3 steps to glycolysis. In the first step, phosphorylation happens by ATP, then it consumes 2 ATP molecules, and thus the glucose is broken down into 2 glyceraldehyde 3-phosphate molecules. Now these 2 molecules of glyceraldehyde 3-phosphate are again processed in the second step where ATP is produced. So, in this first step 2 ATP were produced and 2 ADP were consumed. So, in this step microbes are actually investing their current stock of ATP into glucose, and in the second stage they oxidize this and they create ATP. So, the glyceraldehyde 3-phosphate is oxidized into phosphoglycerate and 2 ATP and 2 NADH are produced. In the third step pyruvate is formed. So, the 2,3-phosphoglycerate molecules are converted into pyruvate and 2 ATP are produced, and overall microbes produce more ATP than they had spent. Now, in case of other sugars such as lactose and sucrose, they are first broken down into simpler sugars such as glucose and galactose, and sucrose is broken down into glucose and fructose. Now, the glucose will pass through the same 3 phases that we talked about. First is consumption of ATP and creation of glyceraldehyde phosphate. Second step would be oxidation of glyceraldehyde phosphate and generation of ATP, and third phase is pyruvate formation and ATP generation. For galactose and fructose, the cell won't let them go waste. So, what cell does is it enzymatically converts them into glucose using isomerase enzyme and then ferments them. Now, I am here showing you an example of a very complicated organic molecule, a polymer rather. So, here we were talking about lactose and sucrose, which are sugars but not as simple as glucose. Here we have a polymer of many glucose or cellobiose chains. So, cellobiose, they look like two glucose molecules that are connected to each other with beta 1,4 glycosidic bond. So, now this is a very complex molecule of cellulose, and you should know that in cellulose, because it is a very long polymer and it is perhaps the most abundant naturally formed organic polymer on Earth's surface. So, the cellulose in itself, in its basic composition, has some regions that are crystalline. For example, here it is flanked by crystalline regions on left side and the right side, and in middle we have amorphous region. So, amorphous region is not crystalline, and as you can notice here pretty clearly. Now, there are 2 broad categories of enzymes that attack cellulose. So, why do they need to attack? Because first of all we need to break down this complex molecule cellulose into simpler glucose structures such as the ones represented here, that can be either fermented or aerobically respired by the microorganisms. So, microorganisms, they produce a suite of hundreds and hundreds of enzymes that act in synergy to degrade this cellulose. So, notice here some of the cellulose are at free end. So, these are non-reducing cellulose, and some of them are connected further, they are reducing end. So, and most of the cellobiose and glucose molecules are in middle. Now, we need different kinds of enzymes to attack different kinds of molecules packed in this structure. So, for the ones that are at the corner, we use exoglucanase. So, these are exocellulase that attack cellulose monomers from the end. Then we have endo beta 1,4 glucanase, which attack cellobiose monomers, which are in the middle and mostly in the amorphous region. So, here CBH2, which is actually carbohydrate binding molecule, they are shown here attacking it from one of the ends, and on the non-reducing end that too, whereas CBH1 is attacking it at the reduced end, and the endoglucanase is attacking it from the middle. Now, when these molecules when these enzymes break down the cellulose, now remember, enzymes do not undergo transformation themselves as they carry on the reaction, and thus we need very little amount of these 3 types of enzymes to break down cellulose into cellobiose monomers. Now, look here, this is a cellobiose monomer. Now, this is the spelling of cellobiose, by the way. Now, they are attached to each other by beta 1,4 glycosidic bond. Now, this bond is a very strong bond, and this is a very stable compound, definitely does not want to degrade. In fact, I want to mention here that cellulose is also one of the most stable sequestered form of carbon, and beta 1,4 glycosidic bonds of its cellobiose may give it immense stability. So, this stable bond requires again a suite of complex enzymes that work together to break it down, and one of the this is actually the rate limiting step of cellulose degradation. So, for breaking down this rate limiting very strong bond between the two glucose monomers which form together cellobios, there is a special suite of enzymes, and they are called then these enzymes, they break down this beta 1,4 glycosidic bond. Now this is very important. This bond is specific to cellulose and different kinds of celluloses, and thus whenever we can find this enzyme, we know that cellulose degradation is happening, and we will see this later in our future lectures, how we use these very specific enzymes as markers, biomarkers, for telling us how much of cellulose degradation is happening and what kind of cellulose degradation is happening. So when these big cellulose polymers have been broken down into cellobios, and then using beta glucosidase enzymes, they have been broken down into glucose monomers, then they can either go into the fermentation pathway, or they can go into the aerobic respiration pathway. Now, because I give you the example of cellulose here, I would like to take a moment to explain to you that the cellulose degradation in itself is a very complicated procedure, phenomena, which involves hundreds of different kinds of enzymes, and they are usually very sensitive to the environmental condition. So for example, I have cellulose degradation happening inside a rumen of a cow. Now you know cow eats cellulose rich food, and it has the ability to degrade it. Unlike human beings, we don't have the ability to degrade cellulose in our intestines, but cows do. So in the rumen they have cellulose degradation going on, and the enzymes they use, the hundreds of enzymes that come together for cellulose to degrade in rumen, are very different and distinct from the enzymes that a wetland utilizes for degrading cellulose. And a simple question might be, why is this cellulose degradation or any such degradation of organic compounds so important for us to study, for our studies to happen? And I would like to mention that this is a hint for you to understand how organic contaminants degrade in environment. Whether I am talking about PHS, which are polyaromatic hydrocarbons, or general hydrocarbons, they all use a suite of enzymes to degrade them, break them down into simpler sugars or simpler compounds that can eventually form glucose or other sugars that can be fermented or respired by microorganisms. One of the chief and highly promoted mechanism for attenuating contaminants in environment is bioremediation and natural attenuation, and both require microorganisms. So it's very important for you to get an idea of what it takes to degrade a major contaminant. Now some of the contaminants, now that we are talking about contaminants, some of the contaminants serve as electron donors and carbon source. However, interestingly, there are also contaminants that serve as electron acceptors and donors of carbon. In some cases, there will only be electron acceptors, and you need to get rid of them. Now this is also a good opportunity for me to explain that most of these mechanisms, as I mentioned just a while ago, are very sensitive to the environmental condition. So if we change the environmental condition, the mechanism of cellulose degradation would change, the mechanism of glucose degradation would change. Now a simple question you might ask, well, what is there to change in glucose degradation? Its fermentative pathway is so well laid out. By the way, after pyruvate is very easy to degrade, so microbes either further reduce it, oxidize it, reduce it into methane, or oxidize it into carbon dioxide and produce more energy. And there's a very nice, very beautiful cycle that happens for pyruvate degradation, but I don't think you need to know this for this particular course. So now the simple question that arises here is that everything about degradation of glucose is known. So we already know the biochemistry of life, we already know how things are broken down, how they are consumed by microbes, but the interesting part is that's not correct. Almost every year, scientists across the world are surprised by the simple diversity in microbial degradation of these organic compounds. Not only are we finding new enzymes, but also new pathways. So every year we add on more information into the metabolism of microbes. Okay. To summarize, there are two forms of respiration, aerobic and anaerobic. Aerobic is more efficient. It produces 38 ATP for every glucose molecule that it respires. Usually performed by eukaryotic cell, it actually is also performed by many prokaryotic cells, so don't worry about that. The glycolysis itself will yield 2 ATP, 2 NADH and 2 pyruvate. The pyruvate can be broken down further. In case of eukaryotic cell, it goes from cytosol to mitochondria, and they use obviously oxygen as electron acceptor because oxygen is available. Now in anaerobic environment, the anaerobic degradation of glucose is relatively inefficient, and it's performed by many prokaryotic organisms and many eukaryotic muscles. As I mentioned earlier, that even human body will undergo anaerobic respiration under dire need. It yields lesser amount of ATP, some ethanol, some lactate or carbon dioxide. It does not happen in mitochondria in case of eukaryotic cell, happens only in cytosol. It does not use oxygen obviously. Both of them require glucose, both of them initially use 2 ATPs, and both of them generate energy and are sufficient for life to move on and sustain. Now, before I go any further, I would like to take few more minutes to explain to you what a chemostat is and how it works, and how we can use a chemostat to optimize cell yield, to make best use of whatever microbial energetics we understand. In previous lectures, I mentioned to you about batch reactors, in which we bring our microbes into a particular media that has enough food for them, enough nutrition for them to grow through their log phase, through the exponential phase, their stationary phase and their death phase. Now, the reason why they die is because they run out of nutrients, and also they produce daughter products that are likely to be toxic to the microbial community. And as such, scientists few decades ago came up with the idea of chemostat. So chemostat is basically a setup where there is a continual input of nutrients and a continual output of my biomass, consumed products and waste material. So a chemostat might look like this. So in chemostat, and I have made the one in which we have the facility to continually stir the media. This is the media, and at the beginning again, like we did in our simple broth, we inoculate it with microbes. So we add microbes here, and microbes find plenty of good food here and they start consuming it. So again, as usual, they go through their lag phase and then hit an exponential increase phase, exponential phase. So in the exponential growth phase, they use these nutrients, and up till here it is very much like what we studied earlier. But now we have changed something. We have a continual inflow of nutrients. So microbes really never run out of nutrients, and because they never run out of nutrients, their exponential phase is pretty good, and once they reach their stationary phase, they sustain this stationary phase for very long. Now this is very important. Once we discovered how to sustain the stationary phase for very long, and this might go very long, I'll give an example. For my dissertation, I maintained this stationary phase for cellulose degradation for more than 200 days, something which can only be maintained for up to a month maximum, and I did it for nearly 2/3 of a year, which is pretty neat, doing it for 8 months. So in simple growth culture, it might have started decaying from here, but in case of chemostat, it will continue in stationary phase for much longer, because then they never run out of nutrients. Now another thing that happens is, for example, in case of cellulose degradation or in degradation of other organic compounds, often the daughter products that are formed. So remember, in cell replication we talked about parent cell and daughter cell. Similarly, in case of organic degradation, we talk about the parent compound or the original contaminant, and then it degrades into daughter compounds. Now some of the daughter compounds have a feedback mechanism in which they inhibit the degradation of the parent compounds, and in case of cellulose degradation, that's definitely the case. So the waste material that they produce or the cell debris that they produce might inhibit the reaction and push it towards cell decay. So not only are nutrients being continually added, but also there is a continuous outflow of the mixed media here, which definitely includes some of the biomass, some of the spent media, some of the waste products and nutrients. Now if we change our nutrients, the inflow of nutrients, we can change how the cell yield would be, how much the microbes can be produced. If we change our output and input, we can also determine how much microbes will sustain in the long run or in the stationary phase. Therefore, chemostat gives us the ability to control two things. It allows us to control nutrients in the reactor, in the chemostat. It also allows us to control how much biomass will be present in the reactor. So I can have high nutrient, low biomass, I can have low nutrient, high biomass, I can have high of both and low of both, any of the combinations I can create within the chemostat. So that flexibility is produced. Now why do we talk about chemostat when I am talking about microbial energetics? Because most of the studies in microbial energetics utilized chemostat. It was very helpful when we could alter the nutrition level. In other words, we could change glucose levels, we could change levels of cellulose and other organic compounds and see how that impacted the microbial community. We could also give microbes a longer duration of healthy time. Remember, we talked about how microbes are healthiest in their exponential growth phase and second best healthy in their stationary phase. So we could prolong these phases, the exponential growth phase and the stationary phase, up to sufficient length that we could study these microbes more deeply and we could understand what enzymes they are making. We could use some of the wonderful tools of microbiology to study these enzymes individually and understand, okay, for cellulose degradation we have two kinds of bonds that need to be broken, and there are two different kinds of enzymes that are doing it. One is exo, one is endo, and then we have the cellulobiomes that are being broken by beta glucosidase, and all this study utilizes the chemostat. So now because we started studying chemostat, we started growing these enzymes we started studying these using chemostat. We started growing these enzymes at sufficient concentration for us to isolate them, to study the genes that actually translate these enzymes, and we found out that it's not just one enzyme that catalyzes step 1 for any chemical reaction, or step 2 or step 3 and so on and so forth, but there are multiple enzymes. So for example, we have sulfate and it needs to be reduced to sulfide form, and this is sulfate reduction, and this is not one step process as I have drawn here. There are multiple phases the sulfur undergoes to go from its highly oxidized form to the highest reduced form it can go to. Now in this we have at every step of sulfate reduction different kinds of enzymes that are working. Now for example, let's talk about dissimilatory sulfate reductase A. This is one enzyme and it can be summarized as DSRA, dissimilatory sulfate reductase A. This is one of the enzymes that catalyzes reduction of sulfite, which is an intermediary between sulfate and sulfide. Even though this enzyme is very well characterized and the gene that makes it or that codes for it defined as DSRA, again similar name. Now this gene is very well known and it is utilized as a genetic marker. Now let me write that down because this is an important term and we will return to it. We know the sequence of this gene. So in other words, we can imagine that we know the structure of this enzyme, we know how it works, but what we are noticing increasingly so is that DSRA is not the only gene that can catalyze this, and we are also noticing that there is a huge diversity within this enzyme. So for example, even in the genetic code there is a diversity, there are slightly differences, different gene sequences that encode this gene, and I am sharing this with you to explain to you a simple reduction of sulfate is not pure chemical reaction, because if you remember, sulfate to sulfide had a plus delta G. And thus catalysis is very important, and there are different catalysts that act together to carry forward this reaction, and that's where the real beauty of bioenergetics comes in. So once you know the basic drivers of bioenergetics, which we talked in this lecture and the previous lecture, which simply is that delta G should be less than 0 for any reaction, and then you know the two formulae for calculating delta G, you will be given the tables, and once you understand and grasp this, and you know how the bigger the reduction in delta G you can achieve by any reaction, the more energetically profitable it is. Once you understand this, then we can proceed and look at the microbial aspect of bioenergetics and understand that how microbes actually carry this energy using ATP, how they mediate the reactions, redox reactions using NADH, and how different enzymes help ATP and to convert into ADP and revert back to ATP. Then we move forward and try to understand the diversity of enzymes that carry on these beautiful biochemical reactions. For example, glycolysis, I presented it as a very simple 3-step chemical reaction, but in your homework for this week, you will explore how beautifully complicated and diverse this pathway is. So in the next lecture, I am going to talk to you about the diversity within prokaryotes, within the microbes, and we will proceed from our current understanding of microbial energetics, how microbes degrade different electron donors, how they use electron acceptors in environment, and we will see how this diversity is a representation of the eminent diversity within the microbes themselves. So that's all for today. Thank you.